Good morning, everyone. Thank you for joining us this morning for the CCH seminar series. This morning, Ihsan will deliver a kind of introductory seminar. He will introduce his PhD project in detail. You can see the title already on the screen. And after that, he will also talk a little bit about his current project at our center. Thank you, Ihsan, for giving us this seminar, and the floor is yours.
Thanks, Levi, and hi, everyone. Thank you for attending the seminar. Before I even start, for people who don't know me, I speak very, very fast, almost twice as fast as you. So if I'm speaking too fast, please let me know. I'll slow down because this is sort of my habit, which I can't seem to change. The other thing is like so as the title suggests, range expansion of Phytophthora species, in particular Phytophthora cinnamomi, into colder environments. So this is sort of a new concept for Phytophthora species, at least, that climate is changing and the cold areas are not that cold anymore. And so this is ongoing debate whether the Phytophthora species are expanding their range. So is it because of the adaptation or is it because of the changing environment? So this is a big question mark. So I'm expecting that we have students here as well. So that's why, Levante just talked about that Roger Schwartz might know Phytophthora, but other than him, I don't know anyone except Adam who knows Phytophthora and natural ecosystem. So it's not about agriculture, it's not about paddock, it's not about like agriculture crop, it's about mountain and natural ecosystem. That's why I have put together the slides in the form of a story so everyone will follow. And I have tried to provide you enough background so it's easier for everyone. But it's new, but it's very easy to follow. It's not like hard. And due to time limitation, I'll be giving you like sort of the blueprint of my thesis, and I have put the publication link against each chapter. So if you want more detail, you can always go and visit the papers if you want more details. With that, I would start. So what's happening? Can't seem to. Oh, this is working. Okay, it's okay, but I can't seem to with the clicker. I can't do that. So this story begins in the early 1990s, where patches of dying Axolobium arborescens shrubs were observed at Bennington Tops National Park. It is in the New South Wales, and its elevation is 1560 meters. So Mills, 1999, he was a PhD student. He went up there and he investigated the cause of the death of these species. So he isolated Phytophthora cinnamomi from the soil beneath dying plants. This is not soil beneath dying plants. So this was a bit surprising because previous surveys have reported that no disease can be found above 440 meters. And as I just said, the elevation of Bennington Tops elevation is like 1560 meters. So this is a huge gap. And also, there's a report that Phytophthora cinnamomi is not pathogenic. It cannot cause disease below 10 degrees C. And the mean minimum temperature at Bennington Tops is 3 degrees C. This means like Phytophthora cinnamomi should not be able to cause disease there. That's why Keith McDougall, this guy, he's my co-supervisor, he said, I don't believe this. And he went up there again. So he said like the presence of pathogen in the soil does not mean this causing disease because it could have been transported in the form of oospores accidentally and they might have detected them. So he revisited the site in October 2001 and he isolated Phytophthora cinnamomi from the roots this time. So this is a proof that sort of Phytophthora cinnamomi was causing disease. Now, why was this discovery important? Like, what's the importance? So for globally, like more than 90% of all color rods of woody plants are caused by Phytophthora species. So this is a huge number, 90%. And only in Western Australia, 54% of species are susceptible to Phytophthora cinnamomi. So only like more than half are susceptible to Phytophthora cinnamomi. So if Phytophthora cinnamomi move to mountain, this can have a very devastating and irreversible effect on the mountain. So this is like a very crucial finding. So, and just a little bit more background. So Center for Phytophthora Science and Management, where I came from, Murdoch University, this is the biggest center in the world, which I've been told, I don't know. So they collected, I think, the most expensive study on the metabarcoding in the world. What they did, they collected 640 samples across Australia. And out of these 640 samples, 110 samples were from the subalpine areas of Tasmania, Victoria, and New South Wales. So they detected 33 species in the subalpine and alpine areas of KNP only. It included Phytophthora cinnamomi at 2100 meters above sea level using HTS techniques. So we wondered, like, what's the reason for the higher Phytophthora diversity in temperate mountains, which were assumed to be Phytophthora free. So this was sort of the aim of my thesis, like whether this is the current temperature gradient. So maybe the environmental condition at higher elevation have changed and they are not that cold anymore. Maybe the pathogen is adapting, or it's the combination of both. So with this, I can go to my thesis. This was the aim of my thesis. So imagine some alien has come to some new area and you sort of doing experiments on it. So if you want to determine Phytophthora species in natural ecosystem, first we need to know which one is the best technique to determine Phytophthora species in the first place. Because if you don't know the best technique for determining Phytophthora species, you might be wasting your time. So first, we need to determine Phytophthora community, and then we sort of survey alpine and subalpine areas to isolate living Phytophthora species for more experiments. And third, we examine the ability of Phytophthora to cause disease and in those conditions, in the laboratory conditions. And lastly, we determine the patterns of Phytophthora species richness, composition, and distribution from the bottom to the top of the mountain. So I will show you one by one like what it means, and it's not that hard. So the first chapter was, and the first aim was, like, as I said, we need to determine the best method for Phytophthora community. I'll give you a blueprint. If you need to find more details, you can always go and visit the paper and find more details. So the reason for the experiment was we needed to accurately determine Phytophthora community in mountain ecosystem. So there are two techniques, traditional techniques and DNA-based HTS techniques, metabarcoding. So traditional techniques are important anyway because you need to have living isolate for experiments. You can't have that with HTS. But do they actually represent Phytophthora community in area? This is the question. And the DNA-based techniques, although they are fast, they can process large sample in a limited time. But which is the best substrate? Do you need to isolate? Do you need to extract DNA from roots, from soil, for bed plants or filters? I'll show you what they are. So the aim was best technique and best substrate determination. So what we did, I and Trina visited like 5 different parks in Western Australia: Bold Park, Kings Park, Etherdale Foreshore, Bibra Lake, and Manning Park. I don't know if you are aware with WA or no, but these are sort of different parks, 5 different parks. So this is my experimental design. It looks a bit crazy, but it's very simple. So we went to 5 different samples, urban bushland samples, and we collected soil. Okay. Now some of the soil, I used the soil and I grow plants in them. This is eucalyptus and banksia plants. And from this soil, I collected some roots as well, washed roots, later on for DNA extraction. And from some of these roots, I infected Phytophthora on apples. So I thought Phytophthora will infect the apples, which apple, and then I would create the infected tissue on the media, and I'll get Phytophthora. And then some of the soil I used in the bedding. So what if we do? People who are not aware of Phytophthora species, we collect some soil, we put water in them, and then we put leaf bed on them. So Phytophthora produce sporangia. Sporangia release zoospores. They infect this host tissue, and then plate them on media, and you get Phytophthora species. And after a week, you filter that water, and you put the filter on the media, and also extract eDNA for HTS studies. And after 3 months, I harvested the plants and I isolated the roots, and I extract eDNA to find Phytophthora. So now I need to know whether Phytophthora, I can get more Phytophthora here, 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 or here. Which is the best technique for Phytophthora species? So this is the sort of comparison of traditional methods, which is bedding, filters, and apple. And this is metabarcoding, which is like soil that filter field roots or glasshouse bed roots. So and I detected a total of 25 Phytophthora species. This is the number of, this is the name of species, and these are their clades. So in case of bedding, I obtained only like 6 species. These are the number of isolates. So Phytophthora meteora, etc. 6 species. In case of filters, again 6 species. And in case of apples, I did not isolate any species. So this is useless. And in case of metabarcoding, I detected 13 species from soil, 24 species from filters, 25 species from field roots, and 19 species from the glasshouse roots. So sometimes people say like DNA can detect dead organism, and that's the problem, you know. So in this case, we actually grow glasshouse plants in the soil, so they would infect the roots, and then we would isolate Phytophthora species. So these are like sort of represent living species. So, and I would like to point out that in some cases, like some Phytophthora species were detected only in one substrate. For example, this was detected on the field roots only. This was detected only in the soil, Phytophthora melanos. So if you have got money, it's always good to have use multiple substrates. So you can see in case of traditional molecular techniques, who is the winner? And in case of the different filters, soil, glasshouse roots, or whatever, soil roots are found best to detect Phytophthora species. And the other thing with the soil roots is which Trina detects. If you try to detect Phytophthora species or whatever, if you do soil versus soil roots, it's always good to have like roots, because roots acts as a biological filter. So if species have to be alive to infect roots, so there's more chance of detecting species in the living species in the roots than soil. So it's always Trina always like to use roots anyway. So with this, the second aim was to sort of survey alpine and subalpine areas to isolate Phytophthora species for further experiments. And also determine baseline species. So what was the background? Well, that's right. CPSM conducted a study in 2013, and they collected 6 food samples and 1 tenor from the alpine and subalpine areas, and they detected 33 species using HGS. But they were not proof of living organism, because she collected the soil, used soil eDNA for Phytophthora detection, and HGS techniques can just tell you Phytophthora is there or not. It cannot tell you like whether it's alive or dead. So it's sort of a big problem. So our aim was to go there and isolate living Phytophthora species. So Keith went to New South Wales and he collected 18 cell samples at elevation range from 1275 to 2228 meter. This is alpine and subalpine areas. And Trina collected samples from Tasmania at elevation range 735 to 1254, 6 samples. So what I did baiting. So this is the same thing which I told you before, like I just wanted to isolate living Phytophthora species. I collected soil, and then I put the soil in a container, put water in them, then I put leaf over them. So the idea was the pathogen would produce sporangia, sporangia would release zoospores, zoospores would infect the leaf tissue. I would plate them on the media, and then I would get Phytophthora. That's all I did. So in the results, I got 8 Phytophthora species, and 2 of them were new species. Okay, so new species, I would just talk a little about them, just a few slides, then I'll go back to the other chapter. So the first species was Phytophthora aureophila. Aureo mean mountain and phila mean living, so mountain living species. This was the origin, Latin word. And if you have to, the following few slides are due to this, if you have to describe a new Phytophthora species, you need to do like sequence different gene regions, and then you do the morphology, you measure sporangia, chlamydospores, hyperswelling, oospores, and then you take photos, and then you measure the temperatures same as other species. So based on the phylogeny of this sequencing these different gene regions, Phytophthora aureophila was close to Phytophthora rosacearum, but there were a bit of a differences. Phytophthora aureophila has a lower optimum temperature. You know, you can see it's 20, and Phytophthora rosacearum has about 30. So this was one difference. Other difference was this is aureophila and this is rosacearum, and these are the name of different medias. Yeah, so they have got different structures on the same media. And this is the some sporangia and oospores and hyperswellings. I don't have the measurements, but you can read the paper and you can see it there. And the other species was Phytophthora curcuminis, which is from a Latin word, curcumin mean pig, so sort of. And based on the implication of these gene regions, it was found closely related to Phytophthora felix. And so this is curcuminous on growth of curcuminous, and felix on carrot agar V8, multi-state agar PDA media. So you can see on the same media, they have got different, on multi-state agar, they have got different structures and different morphology. it has got a specific name as well, which I don't remember now. Okay, so these are the different. I did not find any oospores in this case. I just found sporangia and chlamydia spores, and that's all. I did not find any oospores in this case, so I don't know. Okay, the other point, the last point I'm trying to make here is like Phytophthora species are there, and they're not just there; they are causing disease as well. So this was the first case where we found Phytophthora gregata infected Thymelia bracteata. So the healthy tissues, healthy plants look like this. When they're infected by Phytophthora, they become like stick. And when you sort of examine the root tissue, you can see the typical Phytophthora symptom here. And the elevation is like 1270 meter. It's a subalpine area in Kelly Plains. Okay, the third aim of the thesis was, okay, Phytophthora species are there. Phytophthora cinnamomi has been detected through HTS. Whether it can cause disease or no. This was the aim of this chapter. So if you want more details, you can go to the paper. I'll just give you a sort of a blueprint. So I've been talking about alpine and subalpine areas. So what are these? So if you go to the mountains, so if you start from the bottom, you will see large big trees. The temperature is not that cold. That's called mountain region. If you go up high, then the number of large trees reduce and the temperature also reduces. So they are mainly shrubs like and snow gums. So that's called subalpine. When you go up further, like up high, you cannot find any tree. It's just like small vegetation because the temperature is too cold for trees. That's called alpine areas. So the focus of our thesis was this area. Now I would like to introduce another term because we wanted to determine the adaptive capability of Phytophthora cinnamomi. So phenotypic plasticity. It is the ability of organism to express different phenotypes in a changing environment. What is the example? Example is the development of resistance by Phytophthora cinnamomi to phosphite, because phosphite is the only fungicides which is being used by people to control Phytophthora species. So this study that Phytophthora cinnamomi has developed resistance to that. This is an example. So there are two types of phenotypic plasticity. One is non-adaptive and one is adaptive. Non-adaptive mean sort of shock response. They are not heritable, cannot be transferred to the offsprings. And the adaptive mean the changes are heritable. So the experimental design was similar to which we read in the class in the school biology that that Ivan Pavlov dog, and he would sort of, whenever he would try to feed the dog, he would ring the bell. So after a while, and the dog would produce saliva. So after a while, he would just ring the bell without food, and he would still produce saliva. So we actually stole that experiment and used the same thing, but instead of dogs, so this guy sort of trained the dog to produce saliva with the bill. So in our case, we replaced the dog with Phytophthora cinnamomi, and we wanted to train Phytophthora cinnamomi under cool conditions to grow more, grow faster, produce sporangia, and release zoospores at increasingly lower temperature. Why? To test its ability to adapt to changing environments, in this case cold. And the other aim was, we wanted to sort of determine the ability to we wanted to train Phytophthora cinnamomi to infect and cause disease at increasingly lower temperatures than original isolates. Why? To test its ability to cause disease in changing environment. So we wanted to test its ability to adapt to changing environment, and we also wanted to test its ability to cause disease in changing environment. Phenotypic plasticity. So simple methods. If you want to train some Phytophthora cinnamomi to produce sporangia at lower temperatures, want them to train to infect hosts at lower temperatures, first you have to find out what is the temperature range. So I determine the temperature range for radial growth on agar, V8 agar, and then I found at what temperature it can produce sporangia and release zoospores. And then I found at what temperature, at lowest temperature, Phytophthora cinnamomi can infect host and survive. And then I would train them to sort of increase, produce spores at lower temperature, and then infect host at increasingly lower temperatures. So first I had to have Phytophthora cinnamomi to train them. So I had about 30 isolates. Only one was from the subalpine area, other was from Queensland and Perth and Hart areas. And the radial growth on agar, you can see that the slides are not very good, but the growth range was for ideal growth range was from 10 to 30 degrees C, and the most growth was found at between 25 and 30 degrees C, except only one isolate which was a bit weird. Other was like between 25 and 30 was their optimum temperature for growth rate. And for sporangia production, the range was between 10 and 30 degrees C, and the most sporangia are produced at 25 degrees C in most cases. So that 25 to 30 degrees C, they produce more sporangia, release more spores, and they can grow faster in a nutshell. Okay, now last thing I needed to find out the lowest temperature at which Phytophthora cinnamomi can infect. So what I did, I grew inoculated Axolopian arborescens. This is the subalpine shrub with 4 isolates. I put them in a growth chamber at 8 degrees C, and I put 1 isolate in the glass house as a control. And then I grow them in the glass house like in the growth chamber for 3 months to see whether it can infect and survive or no. So out of 4 isolate, only 1 isolate was recovered. That's 188. So if I would increase the thing, so this is the blue ones are the recovered isolate, and the orange ones are the original isolate. So I tried to produce sporangia and release those spores. I tried to measure that again, and I did not see any difference. They had the same growth, so there was no difference at all. So now we have a sort of idea of the temperature range for growth, which was between 5 to 35 degrees C. The temperature range for sporangia was between 10 to 30 degrees C. The lowest temperature for infection was 8 degrees C. Now it was sort of time to train them to increase, sort of produce sporangia at lower temperature than this, make Phytophthora cinnamomi to infect host at lower temperature than this. This was the idea. So these are the phenotypic plasticity experiments. This was first experiments. So this experiment took like 6 months. It looks very easy, but it's not. So in this case, I inoculated 5 isolates at 9 degrees C and put them in the growth chamber for 3 months. And at the same 5 isolates, I put them in the glass house at ambient temperature, normal temperature. So after growing for 3 months, I recovered the plant, I harvested them, and I sort of isolated Phytophthora cinnamomi, and I again did the cycle again. I made them produce sporangia and reduce those spores. So out of 5 isolates, 3 isolates were now able to produce sporangia at 7.5 degrees C. And initially they were able to produce sporangia only at 10 degrees C. This means that Phytophthora cinnamomi has a plastic genotype, and it can rapidly adapt to changing environment and can cause disease. And not only that, so at 7.5 degrees C, in the original experiment, there was no sporangia. Again, there was no sporangia, but in the phenotypic plasticity experiment, I made them produce sporangia in a very fewer days, 15 days, compared to 24 days at 10 degrees C. So not only they were able to produce sporangia at 7.5 degrees C, but they were also able to produce sporangia in fewer days. Okay, so in phenotypic plasticity experiment 2, I wanted to know whether they can cause disease at lower temperature or no. So as we know that the lower temperature for infection was about 8 degrees C. So in this case, I wanted to sort of make them to infect plants at 7.5 degrees C. So I grow the isolates in the growth chamber at 7.5 degrees C for 3 months, and other same set of isolates was grown in the glass house at normal temperature. But after 3 months, I did not record any isolates from at 7.5 degrees C after 3 months. So it is not clear whether Phytophthora cinnamomi was not able to infect plants at this temperature, or it could not withstand cold conditions for 3 months. So this is still a big question mark. I made them produce sporangia at lower temperatures, but I could not make them infect plants at lower temperatures. So more work is required in this case. So by the way, this is a question, some of you might ask, like, what alpine and subalpine regions fear Phytophthora cinnamomi? This is the question, like, so the problem is this, like, we will never know the whole thing, but previously when they were doing the survey, they would go there and they would check if there are symptoms or no. So Phytophthora cinnamomi presence was based on disease symptoms. If no symptoms, no disease. But there are problems with this approach. The first problem is the presence of pathogen does not mean it's causing disease, because you need to have that disease triangle. You need to have pathogen, host, and environment for a sufficient time to like cause disease. The second problem is Phytophthora cinnamomi can live in the host tissue without causing symptoms. It could be there, but it's not causing disease because it's too cold. The third problem is, since 1940s, when they were conducting the surveys, they were conducting under the assumption that, okay, these cold mountains are very cold anyway for Phytophthora cinnamomi. So they did not even bother, like just sort of, they would check in and just come back quickly. So they were sort of, they had this assumption that they are too cold Phytophthora cinnamomi and it cannot be there. So we will never know. I'm trying to make like, yes, it could be Phytophthora cinnamomi could be there like for a long time, but we would never know the answer, right? And the final question is, they always use the traditional techniques, and we just saw that traditional techniques are not very effective to say whether Phytophthora is there or no. You have to have both techniques, traditional and STS techniques, to determine Phytophthora species. So if you want more information, this is the paper which my supervisor published in modeling paper as well. So it has got the global map of current and projected distribution. You can read that if you want, but we will leave this chapter here. And the last aim of the thesis was to determine patterns of Phytophthora species richness, composition, and distribution along a steep elevation gradient, and compare this pattern with those of vascular plants. So why was this important? So people who are a bit new to ecology, I'm also new to ecology. So the alpine and subalpine vegetation, they are very precious. They don't want to lose them. So they take care of them. So they don't like the weeds. We are pathologists, we don't like pathogens, and they don't like the weeds because they then compete with the native vegetation and it's not good for them. So what they do, so the approaches for managing invasive weed depend on their distribution and abundance. For example, if the weed species are localized, they're local and very rare species, they would just destroy them. If the weed species are too abundant to sort of to be destroyed, they would try to contain them, but it's very rare. It's hard to like sort of contain and restrict weed species. And if they are very widespread, then they would try to sort of protect other areas and try to prevent the spread. So the same approach can be used for Phytophthora species as well. For example, if Phytophthora species are localized, they are locally distributed, they are not widespread, you can either eradicate them, destroy them, or use spray to sort of contain their spread. Or if they are widespread and abundant species, then all you can do is to sort of protect the current trees and prevent further introduction, because once Phytophthora are there and they are widespread, then it's really hard to control them. And on the mountain scale, it's hard to use fungicides and that type of stuff. So we wanted to know, like, what's the story of Phytophthora species? Are they abundant? Are they restricted distribution? What's happening? So as I was new to ecology when I first started my PhD, so I assume most of you are also new to this concept. So I'll just introduce a few concepts, because without this it will be very hard to follow the next few slides. So the basic concepts is this. In protected mountains, for example, CCH-USQ National Park, if you go there, so people just don't go there every day, they are very protected mountains. So there are two concepts. One is road, one is natural vegetation habitat. Road, as the name indicates, they are the roads. And natural vegetation here, it is the far away from road. In our case, it's like 100 meters. The other concept which I'm trying to make is introduced species versus native species. Introduced species, as the name indicates, they're introduced, they're not from there. Native species are there for hundreds of years, and you can visit this website to find which are native, which are non-native for New South Wales. Okay. As I said, introduced species are introduced, so they are introduced mainly via roads, because people just don't go there just for anyway. They're very hard to get in, very hard bushes and very hard protected mountains. So people just go for a visit or something, they use the roads. So introduced species are mainly introduced through roads. I'm talking about plants here. Okay, and as we go up the mountain, the number of introduced species decreases, because why? They are not native, so they don't have the adaptation and resistance to cold condition. So they cannot. As we go up mountain, their number of species decreases, and as we go up mountain, there's less human disturbance also as well. So their sort of introduction decreases anyway. And the other thing is decreasing native species elevation is scale dependent. This means the native species which are here somewhere, they are there for thousands of years, so they are well adapted to cold condition. So it depends on the area. Some areas they decrease with the increasing elevation, and sometimes they don't decrease because they are really adapted to the cold condition there. The third concept which I want to introduce is species composition. So plant species composition is always different on roads, on roads and natural vegetation, because as I said, introduced species are mainly introduced via roads, so they are mainly confined to roads. And natural vegetation, they are mostly in the natural habitat anyway, because when they make roads, they introduce new species. So these landscape barriers and less human disturbance here. So this area is mostly occupied by native plant species, and this area is mostly occupied by introduced plant species. With this, we can probably follow the following slides. So our hypotheses were, same as vascular plants, introduced Phytophthora species will also tend to occur more frequently on road and vice versa, like native species on the native vegetation and introduced species along the road. Okay, the second hypothesis was, as we know that as we go up the mountain, the vascular plant species richness decreases. Phytophthora will follow the same pattern because they are the plant pathogens, so they will follow the same pattern. And the third, we thought, if vascular plants, they occur in discrete communities, I mean introduced species are confined to roads and native species are confined to the natural vegetation, Phytophthora species will also follow the same pattern. This was our sort of thinking when we were doing the experiment. So this was very expensive study, and I would like to acknowledge Mountain Invasion Research Network. They do conduct this survey after every 5 years. Without them, it would be impossible, because we sampled 3 roads in K and P. The elevation range was from 410 to 2125 meter, and they paid for the survey and my sort of residence and everything. I didn't have to pay anything. I used their vehicles and everything, and they were with me. I couldn't have done without them. So each road has like 20 transects. So this is 1, 2, 3, 20 transects. This is the mountain. So this is like sort of up to the top. Each transect has got 2 samples. One is road sample and one is the natural vegetation sample. Now I just said what they do, they lay a 100-meter tape here. So this is 100-meter area complete. This is road plat, which is only 2 meters. So between the road, from the road, only 2 meters. This is the road plat. They call it disturbed. They are mainly occupied by introduced species. And from 50 meter to 100 meter, this is called natural vegetation plat. This is called undisturbed because there is no disturbance. In the road, people go through vehicles or cycles, whatever. This area is disturbed, and this area is undisturbed, natural vegetation. So in case of ecologists, I was with them, so they are very fast people. They would just come here, 2 meter here, and they would just quickly record the number of species here. And they would come here from 50 to 100 meter, and they would just go along the tape and record the species. They are very experienced, and we cannot do that. And they just know the species. They just go along the tape and they record the species. In my case, I collected 5 samples from here, different places, and 5 samples from here. So this was sort of road plat in my case, and this was natural vegetation plat. In their case, it was, this is the road, and this is natural vegetation plat. And I used baiting and STS techniques for both techniques for Phytophthora species. And then we use the GLMs to investigate the distribution patterns. So I detected a total of 25 species there. The species which are sort of shaded, they are recovered through baiting, and the other are through STS techniques. So they are called phylotypes. Okay, now we're comparing the species distribution. First, in case of introduced species for vascular plants, on the roads, they were more recurred than natural vegetation. And as I just said, they are mainly introduced through roads, so there were more introduced species on the roads than in the natural vegetation. In case of Phytophthora species detected through STS, there were 40 records on the roads and 44 in natural vegetation. Now this is very important because Phytophthora comes here and it quickly goes to the natural vegetation. So this is not good for the native vegetation, and they quickly spread. In case of baiting, 4 were on the road and 8 were in the natural, but it was still not significant. In case of vascular plants, there was a significant difference in species richness. Okay, now for native species, in case of vascular plants, there were 861 records on the roads and 1777 on natural vegetation. Why is that? Because they are native, they are mostly in the native vegetation, so there's a significant difference. In case of Phytophthora species detected through STS, 189, 204, no significant difference. 11 and 12, no significant difference. So this is very important. When Phytophthora species go to the mountain, they quickly spread from root to the natural vegetation. So no good. And if we compare their elevation ranges, in case of Phytophthora species, both on road and in natural vegetation, they had higher elevation ranges than vascular plants, both on roads and in native vegetation. So Phytophthora species go there and then they quickly go spread to the top of the mountain, also to natural vegetation. And this is sort of their distribution pattern of Phytophthora species, which had like 14 or more occurrences. So some species in an area, for example, they were more here and then a bit more here and then decreased here and then again decreased. So this is the elevation range and this is the species. So, and the width of the column indicate the frequency. So different patterns. In this case, they were like sort of few species here and then they increased here. So it's interesting. Hard to know why is that. Different patterns here. So now just a comparison of baiting and STS technique. In case of STS technique, the Phytophthora species had higher elevation range than baiting species detected by baiting. So this is again the classic question. They may not be surviving there. They may not be alive there. We never know. Although we detected from roots, but still there's a question mark. And this is sort of the comparison of their species richness, mean number of species. So in case of vascular plants, this is the number of species per plant, and this is the elevation range. In case of introduced species in natural vegetation, as the elevation increased, number of species decreased. Similar as on the road plants, as the elevation increased, the number of species decreased. But you can see the effect was less on road because they're introduced species, they're mainly on the road, so the pattern was not that thing. In case of native species, which are there, as the elevation increased, number of species decreased, both on roads, on natural vegetation and roads. So the point I'm trying to make here is, for vascular plants, whether introduced or native, as the elevation increased, their number of species decreased with increasing elevation. In case of Phytophthora species, which were detected by baiting, we found the same pattern, like introduced species, native species. As the elevation increased, both on roads and natural vegetation, their number of species decreased. So they actually followed the vascular plant species pattern. The species recovered through baiting. But the species recovered through STS, there was no significant difference with the increasing elevation. So as the elevation increased, you can see the difference is not very high. So there was no significant decrease. So again, it's the question whether STS species were, maybe they are not alive, because as the elevation increased, we did not found a significant decline in the number of species richness with increasing elevation. So major findings from the thesis were, if you want to determine Phytophthora species in a mountain, whatever area, the traditional techniques are not enough. You have to have used like both traditional and STS techniques. And if you want to use that, we found fine roots as the best substrate for the STS studies. And the second finding is like alpine and subalpine areas, they contain a complex Phytophthora diversity, and there's a need for further exploration. And as you Levante just said that they just found a Phytophthora here near Toowoomba in the mountain as well. So they will just surprise you. And the third finding was Phytophthora cinnamomi, as we saw, it can rapidly respond to selection pressure and adapt to new environments, in this case cold. So we tested that it can actually adapt to cold and cause troubles. The last two findings were like Phytophthora species, when they introduced to the mountain, they rapidly spread across elevation and disturbance gradients, and they are hard to manage. And they also have like higher elevation ranges than vascular plant species. We just saw that. And in case of vascular plants, as we go up the mountain, their species richness decreases. But in case of Phytophthora species, only detected by STS, they did not follow this pattern. The Phytophthora species detected by baiting, they follow the pattern. So we never know, but Phytophthora species, they have mostly higher elevation ranges than vascular plants, and they're sort of hard to manage. Last minute, I'm just quickly talk about my postdoc project, and it will just take a minute. So we just wanted to find out the role of conidia in the dispersal of Ascochyta rabii. And previously, it was reported that the conidia spread only up to 10 meters, not more than that. And we did some experiments. So this is the agriculture of Victoria. They have recently established an irrigation system, and it can automatically, you can just put in the system that I want 10 ml, and it can sort of maintain that stuff. So we use that to determine the spread of conidia in this case. So we use that. This is the system. This is the photo of the irrigation system. So we use like 3 events, 10 mil each. In case of natural rain, we had like 0.8, 4.6, and 18.6 mil rain. So we wanted to study how far they disperse. And in every event, conidia spread up to 75 meter. And of course, the number of lesions were higher near at 0 meter. And then number of the spores lesions detected decreased as this increased. So less than 75 meter. But in every case, the point I'm trying to make that we found the spread up to 75 meter in each spread event. And if you want more details, there's a lot to that. There's no time, but you can go and read the paper here, and if you want interested. And now, the experiment which I'm currently working on, it just aimed to develop a spatio-temporal model to predict chickpea risk. So what I'm doing here, so I'm just putting a single infested seedling in the center of the plot, and I'm trying to determine the spatial spread in all 4 directions and temporal spread at different intervals. That's all. Thank you so much.
Thank you very much, Ihsan, for this very clear, very structured seminar. Very interesting topic. I have a number of questions, but let me ask first the audience if anyone has any questions or comments. Well, while people are thinking, let me start with a comment about your isolation technique versus the metabarcoding.
Mm-hmm.
A very interesting table at the beginning where you clearly showed that isolation of living cultures. I don't think so. I think it's not. This is the one. Previously, you had, I think, a table about that. So isolation techniques, the classical techniques are obviously not as useful as metabarcoding. You also mentioned there that you had plants grown in the glass house and you did metabarcoding from their roots. My question is, have you tried to isolate Phytophthora from those plants grown in the glass house?
From these plants?
I don't know which is the table. Maybe if you go a little bit backwards. Okay. Yeah, even backwards, where you have the number of strains isolated from plants versus number of species shown. Maybe that's it. Yes, that's it. The comparison table. You have their glasshouse bait roots.
Yes.
So have you also tried to isolate from those roots?
Yes, I did. But I only did it from the plants which were dead. So I only isolated only one species, and that was Phytophthora multivora, which should be here, somewhere here.
All right. All right.
Yeah.
So yeah. So you try to do that. It's a very clear study, I guess. The classical technique versus metabarcoding. Yeah. So I really like this point. Another question. So I just wonder how were these Phytophthora species originally introduced to those high altitudes? And
yeah. Okay. So there's a number of papers on that. So since European colonization, they were making the roads for K and P. So it goes about 100 or 200 years back when they were trying to make roads to go up there. And with that road making, it's mainly introduced via those road making time.
I understand. What came in my mind is that in national parks, there are sometimes reforestation projects when native trees, young trees, grown in nurseries, are taken from the nursery to the national park and planted there with soil, obviously with soil. So we know from Thomas Young's papers, you cited those papers at the beginning.
Mm-hmm.
So from his papers and also from a number of other papers,
Mm-hmm.
Phytophthora can be introduced to a number of places with soil taken from nurseries.
Yes, that's right. And there's a big paper on that. And the paper I think I'm setting here as well, somewhere here.
Yeah.
It is Thomas Young paper which I'm talking about is published in 2016, and it has a very big paper on that. But maybe in our case, we are talking about, I've read that paper. In our case, we are talking about protected mountains. So these are sort of mountain which they want to protect. So they would never transfer from, never try to grow plants. In case of roads only, when they try to make roads, so just to prevent soil erosion, they would sort of take plants from nurseries to there, but normally they don't do that in protected mountains. I'm aware of that paper. So you are right about that. But in protected mountains, which is like CCH-USQ National Park, mostly species are introduced through disturbance and just on the roads only, as you said, like from nurseries when they try to prevent the soil erosion. Because when they make the road, the soil sort of disturbs the whole thing. They try to sort of transfer the plants from nursery to the mountain, and that's also a reason for the introduction. I know that. So with that, you are right, but at very high elevations, and especially in the natural vegetation, they spread from roots to the natural vegetation through animals or whatever, through soil movement or through waterways, mainly through that. So on the roads, you are right. And in the natural vegetation, they mainly move through the animals through streams, water streams.
Yeah, of course. Creeks do spread Phytophthora. So it cannot be from nurseries with native plants, you know?
No, not native plant, because if you go to the CCH-USQ National Park and you will see the board everywhere, you know, please protect our flora, and they are very unique. So they don't want to they don't want weeds either. So every 5 years, the Myron group, they go there and they record species to check on the weeds and everything. So they don't like weeds either.
Okay, all right, thank you. I think someone else had a question in the meantime.
Sure.
Please go ahead. I only see the U number there on the screen. U800 0731. I don't know who is this person. Okay, because it's unmuted.
It's me.
Okay, any questions, any comments, please? Okay, no questions or comments. Anyone else? I think that you should give another seminar, Ihsan, about your current project, the Ascokaita work, which is really interesting, and I'm very happy to see that it has already been published. That's a very good outcome of that project. If no one else has any more comments, okay, already had the same question. Okay, already had the same question about the uphill movement of pathogens. So that this was answered by Ihsan. I'm just checking the chat if anyone else had any comments through Zoom chat. No, nothing there. In that case, I would like to thank you once again, Ihsan. It was a really nice presentation, and thank you very much for introducing your PhD topic and also your current project at CCH. Thank you everyone for attending this seminar. It looks like it will be available also on YouTube, on Open Plant Pathology YouTube site. Adam may confirm this. I'm not entirely sure if this is the case. Let's see, Adam is still muted. Yes, it will be available also on YouTube. So thank you, Ihsan. Thanks everyone for joining us. Have a nice day.
Thank you.
Bye, everyone.
Yeah.